Hey everyone, this is Ishmael here from the crew program. So what we decided to do was to share some of the plant identification courses that we've had over the years with our volunteers, to share that on our social media platforms, just to share the knowledge and to create a um, inspired view of plants and to help people identify plant species. So the information I will be sharing is really very basic information on plant identification. We'll start off with just the very basics of flower morphology, and then we'll go on to taking each of the plant families and working through the floral morphology and some of the diagnostic characters that you need to look out for when you're trying to identify these species. So this is the first video we will share. This is the basics of botany, and it's focused on the flower morphology. Hi there, so back to the basics of botany. So this presentation is going to be very short. We're just going to look at the flower morphology and the characters. Just to start off, there's basically two groups of plants that you find, the gymnosperms and the angiosperms. The group that we're most interested in is the angiosperms, the flowering plants. So angiosperms include the groups which have flowers and seeds that are usually enclosed within an ovary. So if you think of most of the flowers that you see out there, gymnosperms on the other hand are things like pines and conifers that don't have flowers and that have naked seeds. And then angiosperms are broken down further into two main groups, mainly the monocots and the dicots, the monocotyledons and the dicotyledons. And these divisions are determined by the number of cotyledons found in the seed, the number of seed leaves. So if you imagine planting a seed, and I'm sure all of us did this experiment when we were at school, where you took a bean or a millie and put it between some wet cotton wool and stuck it in the fridge. Basically the bean, once it started growing, had two little seed leaves that came out, and the millie would have just one. So those are the key differences between the monocots and the dicots. So just to go into further detail, and this is really important when you start identifying plants, that you understand in what group the plant that you're trying to identify falls into. So monocots have one cotyledon versus two in dicots. The floral parts are in threes, as opposed to floral parts being in fours or fives in dicotyledons. Most monocots have parallel leaf veins, and the dicots have net leaf veins. And this is something that you probably won't ever see unless you have a fantastic microscope, but the pollen grains in monocots have one pore or furrow, and in dicots they have three pores or furrows. And then the vascular bundles, of course, also you won't be able to see this with the naked eye, but it's good to know it. The vascular bundles throughout the stem, vascular bundles occurs throughout the stem's ground tissue, and then in dicotyledons, the stem's vascular bundles are arranged in a ring. So these are the two, or some of the main differences between monocots and dicots. So going to the parts of the flower. So you can think of flowers as a set of four whorls or layers. So the outer layers are the sepals, and then the inner layer, and those are usually leaf-like, and they sit on the outside of the flower. So whatever flower you have, if you look at the structures, there's basically four layers. So the first layer is the sepals, and then the next part, and the sepals, one of the functions of the sepals is really to protect the flower and the seeds inside. Then you have the petals, which is usually the brightly colored part of the flower, and then you have the next whorl is the male floral parts, and that consists of an anther, which contains the pollen, and then the filament, which holds up the anther, and together these two things are called the stamen. That's the male parts of the flower. And then In the very center of the flower, you have the female part, which is the ovary, the style, and the stigma. And the stigma is like a receptive platform for pollen to fall onto, and the style transports the pollen down to the ovary where reproduction takes place. And these three together, the stigma, style, and ovary, collectively is called the pistil. We're back, and now we're going to have a look at actual flowers, and I've dissected a few specimens over here so that you can see all these different parts, the sepals, the petals, the male parts and the female parts. Okay, so we're basically going to do a little pelargonium. So there's a close-up of the pelargonium. It's not very clear. So that's what we're going to dissect. And we are going to dissect a little aloe flower. And the reason why I chose these two is that we have an example of a dicotyledon plant and a monocotyledon plant. Okay, so what I've done is I've dissected these flowers, and this is just to show you the different parts. So remember I said that flowers are basically four layers. So this first layer is the sepals, and the sepals collectively are called the calyx. They're usually green and leaf-like this. So there you can see that's the first layer, the first wall. And then if you look carefully over here, you can see that the next wall is the petals, and there I've separated the petals out. So you can see these are usually brightly colored, and the flower part. And then you have the male part of the flower, the stamens, and the stamens is made up of the filament and the anther. And then over there in the inside, you can see the female part, which is right in the center. You have the style and the stigma and the ovary, and I'll show you in a moment what that looks like further. Yeah, so there I've separated out the different parts. So you have your sepals, then your petals, and notice I said that monocots and dicots, one of the main differences are that monocots have flowers in 4s or 5s, and here you can see there's 1, 2, 3, 4, 5 petals, and you have your sepals, and then that's the male part of the flower, the anthers and the filament, and there you have the stigma and the style and the ovary. Okay, moving on to our next flower, we have an aloe flower. So here you can see the actual flower. This comes over aloe. Aloes are monocots. And there's one interesting thing that we need to notice here is that if you look at the flower here, you can see that there's still this outer wall and then an inner wall, right? But they actually look the same. So when this happens, they are called tepals. So tepals are when the sepals and the petals kind of look the same and they cannot be differentiated. But in this example, you can see that clearly this one is sitting on the outer wall. So this is one of the outer tepals, and then this one inside is the inner tepal. And then of course you have your male parts of the flower and then the female parts of the flower. And yeah, I've just separated it out. So here you can, this was just to illustrate the different walls. So here they can see outer, inner, and although they all kind of look the same and they look like a tube, if you take them apart, you will actually find that these are in separate walls. So that's outer, inner, male parts, female parts. Okay, and there's the part separated. And as you can see in this one, in this example here, monocots have floral parts in 3s. So they have 1, 2, 3 outer, and 1, 2, 3 inner. So that's 6 parts altogether.